Welcome back to my presentation on industrial biotechnology. If you look at the basic this microbial growth kinetics, so we have substrate, then we have cell, it produces more cells, and if you have other than cells, you have product, then you have product. Now, in case you have I told you before also, in case you don't have product other than cell, then this we can consider as a autocatalytic reaction, because it is a very good example of autocatalytic reaction. Now, here when you cell, when I told you the cell has the different characteristics, because living cells as the different characteristics. First, the important characteristics is that their reproduction, and second characteristics is they are very sensitive to the environment. Third characteristics is that they are acclimatized to the they can be acclimatized with the different conditions. As for example, that suppose we initiate the organism may not be growing at a certain phenol concentration, like I personally work with that Pseudomonas putida, and when you use the Pseudomonas putida for the phenol degradation, we find initially we find the small concentration of phenol will not allow this organism to grow, because it has some germicidal property. But when you acclimatize this organism with a little bit higher concentration of phenol, then we find that is good for the degradation of even high concentration of phenol. So, acclimatization is a very important property of the microorganism. Then any living system, they also discharge some kind of waste product. Every living system, they discharge some kind of waste product. That is also very important thing that we have. And another thing is that this living system, particularly this microbial system, the specialty is that I told you before also, from one particular state we can get n number of products. We just change the organism, and we can have, depending on the characteristics of the organism, we can get the different products. I have given the example of the glucose. We can convert it to ethanol with the help of Saccharomyces cerevisiae. Same glucose can be converted to citric acid with the help of Aspergillus niger. Now, when we handle these microorganisms, we come across two different terms. One is called doubling time, another is called generation time. Doubling time means time required double the cell population, double with respect to number, double with respect to mass, whatever is there, that is called doubling time. The generation time is basically that it is the time required for the cell division. Not necessarily the generation time and doubling time, they should be close to, they will be equal. It might be different, because it depends on the cell division characteristics. I can give the example. When cell divide, it may divide into 2, it may divide into 3 also, it may divide into 4 also, because it depending on the characteristics of the cells. This is the cell, how they can divide, that is cell division. Now, if one cell divide into 2, then doubling time is equal to the generation time. This is what we have written here. When divide into 2, this doubling time is equal to generation time. Now, before we go for handling this microorganism, it is very essential to know the growth cycle of the organism, because why it is very important, because until unless because the organism basically a black box for us. We don't know how it is growing, what is the status of the microorganism. So we should know the status, then and only then we can handle the microorganism in a proper way. So I can because I shall explain this life cycle of the cell, how it influence the microbial production process. As for example, here you see that this is the life cycle, this is the lag phase, this is the log phase, this is the stationary phase, and this is the death phase. Now, every phase has some significance. Lag phase is considered as the acclimatization phase, because every organism required some time to acclimatize with new environment. So this is usually considered as a non-productive phase. So any kind of organism, this phase should be as minimum as possible. I can give the typical example in case of Saccharomyces cerevisiae, that if the growth media doesn't contain biotin, then lag phase will be higher, which is undesirable. But if the media contains biotin, then this will be reduced. That is a desirable character. It's the same, then the time required for the growth of the organism will be reduced to a great extent. Now, second is considered as the log phase, that what you call as the growth phase. This phase is very, very important, because why it is important? Because the organisms, they remain active at this phase. Whenever we do any kind of inoculation of the cells, this phase we have to take into account, because usually the inoculation of the organism is done either from the mid log phase to late log phase. This is the mid log phase and this is the late log phase. So in between, so we ensured that whatever organism that inoculate, it should be 100% active. Otherwise, you will get the kind of change of characteristics of organism. Then we have some decline phase here, and then you switch over to the stationary phase. The stationary phase is also signifies as the starvation phase. Why the stationary phase occurred? Because here rate of death of the cell is equal to rate of growth of the cell. That is why it is constant. And why it is occurring? Because due to the endogenous respiration of the cells. What is the endogenous respiration? Why it is take place? When your substrate, amount of substrate present in the media is drastically reduced, then the organism, the strong organism will try to use the weak organism for their growth and metabolism. So here, that is why rate of death is and rate of growth there, and and most of the secondary metabolites formation take place in the fermentation process during this stationary phase. The stationary phase, the cell physiology is totally different as compared to growth phase. So since it is the physiology changes, we get the secondary meta, and most of the antibiotics, I can give the example, as for example penicillin, streptomycin and all these things, that is usually produced in the stationary phase. And what is our objective? How to extend this stationary phase. If we extend the stationary phase, then we will get more secondary metabolites. How we can do that? We put the little substrate to the fermentation media, so that the stationary phase may be extended, and we increase our secondary metabolites formation. Now, these are the phase, and this is the death phase. This phase, the rate of death is more as compared to rate of growth, so we are not interested about this phase. Now, for this, the growth characteristics of the organism, that is usually expressed with the help of equation similar to Michaelis-Menten expression, because during enzymatic reaction we discussed about the Michaelis-Menten equation, where V equal to V max S Km plus S. Now, in case of the microbial cell growth, we use similar type of equation. This is mu equal to mu max S Ks plus S, where mu is the specific growth of the cells. Now, what is mu? Mu is 1 by x dx by dt. What is x? X is the cell mass concentration. Now, it can be defined as the rate of cell mass formation per unit cell mass concentration. 1 by x is the per unit cell mass concentration. So, why the specific growth rate means rate of cell mass concentration per unit cell mass concentration. This is what is now Monod equation is mu equal to, Monod equation is what? Monod equation mu equal to mu max S Ks plus S. Now, I can simultaneously I can write the Michaelis-Menten equation here, so that you can find the similarity. This is equal to V max S Km plus S. So, now here this S and this is is the considered as a, this called the substrate. Now, in case of enzymatic reaction, the your substrate is very specific to the enzyme. Am I right? So, as for example, that in case of glucose isomerase reaction, the substrate is glucose. But in case of microbial growth, this is considered as a growth limiting substrate. Growth limiting substrate. Now, what do you mean by growth limiting substrate? That means you see that suppose you have mu and S, it is like this. So, up to this, as you change your S value, your mu changes, and after that is remain plateau. So, above this is independent, mu is independent of S concentration of S, but below this value your mu depends on the concentration of S. Now, if you I told you in the medium, in the growth media, you have different components. You have carbon source, you have nitrogen source, you have minerals, and you have vitamin. So, different components are present in the media. Now, suppose you keep all materials in excess, and concentration of C carbon you change from 0 to infinity, and try to find out that whether the mu changes with respect to carbon source or not. And if you find that it changes with respect to carbon source, we call it growth limiting substrate. Now, suppose we want to find out the effect of nitrogen, we keep carbon, mineral and vitamin excess. So, that excess means the whatever required in the media, we keep it in excess, so that there should not be any, it should not be limiting factor for the growth of the organism. And concentration of nitrogen source you change from 0 to infinity, and if you find your mu value changes with respect to the concentration of nitrogen source, then we call it the growth limiting. Now, if mu value does not change with respect to nitrogen source, it remain constant, then it is not a growth limiting substrate. Same thing happens to the minerals and vitamin. So, what I want to emphasize here, in case of enzymatic reaction, S is the substrate with respect to enzyme, which is very specific, but in case of microbial system, the S is the growth limiting substrate, and it can be anything. It can be carbon source, it can be nitrogen source, it can be minerals, it can be vitamin, anything that we have. So, this we shall have to keep it in mind. Now, we have come across in this in the Ks is the saturation constant. You can see here is the Ks, that is the saturation constant. Now, that significance of the Ks is that if Ks is high, requirement of the limiting substrate is high to achieve the saturation state. That is the, because that is very simple. If you mu equal to mu max S Ks plus S. Now, if Ks is high, then we require more substrate to get the desired amount of cell mass formation. So, if Ks is low, that means you require less amount of substrate to get the desired amount of growth of the cells. Now, what are the limitations of the Michaelis-Menten equation? That if S is infinity, suppose S is in infinity, then I can ignore this with respect to S, and then mu will be tends to mu max. This is what exactly written here. When S is tends to infinity, mu tends to mu max. Now, another thing is that if here mu max and the Ks are the constant. Now, if S is finite, it has the finite value, the mu also should have the finite value. So, what I have written here, S is finite and mu should be finite. Now, it does not explain the situation where what will happen when S tends to 0, because that situation does not explain here, and this equation also does not take care the death of the cell. And whenever we handle any kind of living organism, then we know in a population always there will be some growth of the cell and there will be death of the cells. So, that is the normal situation that we have. Now, that the death of the cells we have not the the monod, they did not consider the death of the cells. Another thing they also they did not consider, that is inhibition effect of substrate and product, that is quite common to the microbial system, that if you keep on increasing the substrate concentration, after some time the growth rate will be hindered. And if our product formation increases, then also we will find that after some time the growth of the organism hampered. So, this equation does not take care of this. These are the limitations of the monod equation. Now, if you want, how you can calculate the rate of growth of the cells. Now, this is the batch process. We know batch process, we take the media, then we take the cell. Now, again I am telling you that when you do the inoculation, we should do the inoculation of the cells in mid log phase, in between mid log phase to late log phase. Then and only then we can get the same results, desired results. Otherwise, sometimes we get some kind of erroneous results. Now, here in the batch process, there is the input and output that should be equal to 0. So, rate cell formation, we can write Rx into V. Rx is the dx by dt, and dx means cell concentration per unit volume. So, you consider the whole volume, so you multiply it by V, and this is the rate of death of the cells in the whole volume, that you multiply it by V, you will get the total cell, how much cell is dying in the whole volume, and this is the rate of accumulation of the cell in the system. So, this you can, if you rewrite, you will come across this equation, that is 1 dx by dt. This is the rate of cell mass for accumulation in the system, is equal to mu minus kd. Kd is the specific death of the cells into x. If the specific death is negligible as compared to the growth of the cell, then we can write 1 by dx by dt equal to mu into x. Here you see, this is if k you considered as a 0, no death of the cell, then equation will be dx by dt equal to mu into x. Then mu will be 1 by x dx by dt. Now, in batch growth, the mu remains approximately constant and approaches to mu max. This is like this, and we can if we assume mu is constant, particularly if we assume that your organisms is growing in the log phase where the rate of growth is more or less constant, then we come across this the equation, that we can integrate that x0 is the initial cell mass concentration, this is the final cell mass concentration, and this is the ln x by x0 equal to mu into x. So, x equal to then if you do this one, then we can here we can easily write, if you write ln x by x0 equal to mu into x, mu into t, then x we can write equal to the x0 into e to the power mu t. This we can write. This is the cell mass concentration at any time t, we can easily calculate. So, t also we can calculate like this in other way. If you see here, t also you can take the mu at the denominator, you will get that. And then doubling time is the, as you know, that doubling time is what, when the cell will be doubled. That means x will be 2x0. Initial concentration will be x0, then final concentration will be 2x0. Then x0 will cancel, ln 2. So, doubling time is nothing but ln 2 by mu. You can easily calculate. Now, question comes, what is the minimum doubling time? Minimum doubling time will be when mu ln 2 by mu max, because the mu max is the maximum specific growth rate of the cells. If you divide by that, we will get the. So, any problem, suppose I give you this, it is mentioned that minimum doubling time. So, you can easily find out the value of mu max of the cells. That is very simple. And then for the how you calculate the generation time? Generation time, you do not know how much cell is produced. Let us assume x0 is converted to xn, then this is xn by ln xn by x0, and mu into gn, this is t gn is the generation time. the d generation time equal ln xn by x0 divided by mu. So, we can calculate both the doubling time and the generation time with the help of this equation. Now, here because I was talking about the specific growth rate can be defined as mu equal to rate of growth of the cell per unit cell mass concentration. Now, similarly, specific substrate consumption rate, how we can express that ds by dt, ds by dt is the rate of substrate consumption rate per unit cell mass concentration. Then it is called a specific substrate consumption rate. Specific product formation rate, dp by dt is the product formation rate, and if you want to add the term specific, then per unit cell mass production, per unit cell mass, how much is the product formation, that is called specific product formation rate. Now, y x by s, the yield coefficient, cell yield you can calculate is the x minus x0, s0 minus s, and product yield you can calculate p minus p0, s minus s0. And this is Monod equation, we can put this, then dx by dt we can write this way. And rate of substrate consumption you can easily find out. How we can find out the rate of substrate? Suppose if I take this equation as the account, dx by dt equal to mu max s ks plus s into x. Am I right? Now, how you can find out the ds by dt? So, I can write the ds by dt equal to ds by dx plus dx by dt. So, dx by dt is equal to what? I can write the dx by ds by ds by dt. No, sorry, this is not like this. Then I can write this equal to ds by dt is equal to 1 by yx by s, because this is equal to dx by dt. So, you can come to the denominator, and this is dx by dt. So, so if you multiply it, that means ds by dt minus ds by dt, you will be what? This is equal to just you multiply it 1 y x by s mu max s ks plus s into x. That is exactly we have done here. You can, this is the equation, we can easily find out. So, then we have shown you during the enzymatic reaction kinetics how to find out the kinetic constant like Km and Vmax. Similarly, Km and Vmax here also with the help of Lineweaver-Bach plot, Addy-Hupstein plot or Henson plot, we can find out the value of Ks and Vmax. Now, let us go to the chemostat process, but let me tell you what do you mean by chemostat. Chemostat means it is basically, this is considered as a CSTR. In the chemical industry, we use the term the CSTR, continuous start tank reactor. When that same reactor we use in the biological process, we consider as a chemostat. The use of continuous cell bacterial growth in a desired phase, that is that we call is chemostat. Now, assumption is that fresh sterile feed x equal to 0 is feed completely mixed and aerated, liquid volume V, steady state, this control the pH, we control the pH, dissolve oxygen and temperature. And then the dilution rate, what is the dilution rate? I told you before also, dilution rate equal to F by V. What is F? What is the unit of F? Volume per unit time. Am I right? And what is the V volume? This is volume. The volume will cancel. The unit is 1 by time. The time inverse. The D unit is time inverse, and 1 by D is nothing but what we call hydraulic retention time. Hydraulic retention time means how long a particular liquid, suppose this is a start tank reactor, if we pass the liquid, this is at the flow rate F, and volume is how long this liquid, particular liquid resides in the reactor. And why we are interested for that, because the time it resides in the reactor, that is the time of reaction. That is why the more hydraulic detention time, that means the more reaction. Less hydraulic detention time, less time we will get for the reaction. Here for the growth of the cells. Now, here how we can analyze the chemostat process, in case of cell balance, we have this is the input and this is the output of the cells. So, this is the input, we can write the F into x0, and then this is the generation of the cells. Then what is the output? F into x, and what is this is the rate of death of the cells, and this is the rate of accumulation. Now, under steady state condition, rate of accumulation is equal to 0. So, this equal to 0, and also if we assume the rate of death of the cell is 0, then also we assume there is sterile feed. Sterile feed means there is no cell present in the incoming liquid, incoming media. Then what will happen? This will be equal to this. This will be equal to this. So, if you multiply it by this, then this is I can write here, so that everything will be clear. Rx into V equal to F into x. What is Rx? Rx equal to dx by dt. Am I right? Into V. Now, we know mu equal to 1 by x dx by dt. So, I can write dx by dt equal to mu into x. So, we can what we can write here, dx by dt equal to mu into x. So, this is equal to mu into x. Am I right? So, here x will cancel. So, I can write mu equal to f. This is V, F by V, that is equal to dilution rate. So, here that means mu equal to d. This is a very important thing. This is possible in case of sterile feed and under steady state condition. So, this is the prerequisite for getting this mu equal to d. Now, why it is required? That let me tell you that here, that if you look at the life cycle of the cells, this is viable cells and this is time, we have this. The rate of growth maximum is this is the log phase, maximum growth. Now, major drawback of the batch process. What is the major drawback of the batch process? Is that you cannot hold the different phase. This is the lag phase, log phase, and stationary phase, death phase. You cannot hold the phase for longer period of time. So, as the time passes on, one phase will be switching over to the other phase. That is the major drawback of the batch process. Now, in case of continuous system, so suppose we have maximum, we want to produce Baker cyst. In the Baker cyst production, we are looking for a high amount of cell mass production. So, we are actually interested to grow the cell in the log phase. So, we have found that mu equal to, under steady state conditions, sterile feed, mu equal to d. Just simple by controlling dilution rate, it is possible to maintain the log phase for infinite period of time. And if we can do that, we can have the maximum amount of cell mass production. Now, here we try to do the material analysis, substrate balance here. This is rate of substrate input, rate of output, this is the rate of substrate disappearance, this is rate of substrate accumulation. But under steady state condition, this will be equal to 0. Then we can come across this equation, d into S0 minus S equal to this. We can easily calculate, and then we can apply the Monod equation, and then mu will be substituted by this, and this called monod chemostat model. This is called this is called monod equation, and this is called monod chemostat model. Now, rearranging this equation, if you here let me tell you that mu equal to mu max is Ks plus S. Now, under steady state conditions, sterile fed mu equal to D. So, I can write D equal to mu max S Ks plus S. So, S you can easily calculate. S you can, how you can calculate? This is equal to Ks mu max minus D. Now, y x by s equal to what? Y by x by s equal to x minus x 0 divided by s minus s 0. Am I right? Now, so I can write x equal to x 0 plus y x by s into s 0. This is not 0, this is s. This is s 0 minus s. So, this is sterile feed, this will be equal to 0. So, if we calculate the value of this, we can put it here, we get the value of x. Now, we can put the value, we can get the value of x. Now, here what is the cell productivity we get? Cell productivity is what if you multiply it, what is the cell mass productivity? Is the dx by dt. Dx by dt equal to what? Mu into x, and mu under steady state conditions, sterile feed mu equal to d into x. So, this is considered as that cell productivity. This is like this. So, and this can be calculated like that. To obtain the now I think this I shall discuss in the next part of my lecture. Thank you very much.